Hey everyone, and welcome. So let's talk about the anatomy of flowering plants chapter. If you've ever found this one kind of tricky or a little bit intimidating, you are in exactly the right place. Today we're going to break down this super important topic for your Class 11 and NEET exams step by step so you can really get a grip on the amazing world hidden inside every plant. So let's kick things off with a big question. We see plants all around us every single day, but have you ever really stopped to think about how they build that complex world inside of them? How does water get from the deep in the soil all the way to the top of a huge tree? And how do they manage to stand so strong against wind and rain? Well, the answers to all of that, they're found in their anatomy, in their internal structure. Let's dive in and see how they pull it off.
So here's the game plan. We're going to start with the basics, the absolute fundamentals, the plant tissues. Then we'll see how those tissues get organized into bigger systems. From there, we'll get into the nitty-gritty of root, stem, and leaf anatomy, really zeroing in on those key differences between dicots and monocots. After that, we tackle the big one, secondary growth, and we'll wrap it all up with an exam-focused recap to make sure it all sticks. Let's do it!
It's like you can't understand a building without knowing what the bricks are made of, right? It's the exact same idea with plants. To really understand the whole organism, we've got to start small, with its most fundamental units, the tissues. And a tissue, well, it's just a group of cells that come from the same place and work together to do a specific job.
First up are the meristems. Think of these as the plant's growth factories. These are the spots where cells are constantly dividing. You've got apical meristems right at the very tips of roots and shoots, and their job is to make the plat longer. That's what we call primary growth. Then there's intercalary meristems. These are found in grasses, and they're why grass can grow back after a lawnmower or a goat comes along. And finally, you have the lateral meristems, which make the plant grow wider or increase its girth. That's secondary growth. So what happens after those meristematic cells divide? Well, they grow up, they mature, they specialize, and they lose that ability to keep dividing. They become permanent tissues, the real specialists of the plant world. And we can split these into two main groups. Simple tissues are, well, simple. They're made of just one type of cell. That's your parenchyma, collenchyma, and sclerenchyma. On the other hand, complex tissues are made of a bunch of different cell types all working together as a team.
Okay, pay close attention here, because this table, it's absolute gold for your exams. It lays out the key differences so clearly. First, parenchyma. These are living cells with thin walls, and they do the heavy lifting for things like storage and photosynthesis. Next, collenchyma. Also living, but check out their cell walls, they're unevenly thickened at the corners. This gives flexible support to growing parts, like a young stem that needs to bend in the wind. And finally, sclerenchyma. These cells are actually dead when they're mature. Their walls are super thick and lignified, making them incredibly tough. Their job is rigid support. Think of the hard shell of a walnut or the gritty texture in a pear. So the bottom line is living versus dead, thin walls versus thick, and flexible versus rigid support.
All right, on to our first complex tissue, xylem. This is basically the plant's plumbing and its internal scaffolding all rolled into one. Its job is to move water and minerals from the roots all the way up to the leaves. Now, it's made of 4 different cell types, and here's the kicker. 3 of them, the tracheids, vessels, and xylem fibers, are dead at maturity. Their walls are thick and strong, which is perfect for transport and support. The only living part is the xylem parenchyma, which just hangs out and stores food. Now, here's a little something to tuck away in your brain. The arrangement of this xylem is different in stems and roots. We'll be talking about endarch and exarch very soon, so keep that in the back of your mind.
So if xylem is the water pipe, phloem is the food delivery service. It moves all the sugary food made during photosynthesis from the leaves down to the rest of the plant. What's really cool about phloem is the relationship between the sieve tube elements and the companion cells. See, the sieve tubes do the actual transporting, but when they mature, they lose their nucleus. So the companion cell right next door, with its big nucleus, has to run the show for both of them. They're basically a team. One can't function without the other. The other parts are phloem parenchyma for storage, and phloem fibers, you might know them as bast fibers from things like jute or flax. Now, here's a classic bit of NEET trivia you absolutely need to know. Phloem parenchyma is missing in most monocots. Don't forget that one.
Okay, so we've got our individual bricks, the parenchyma, the xylem, the phloem. Now it's time to zoom out and look at the blueprint for the whole building. We're moving on from individual tissues to tissue systems, which is how these tissues are grouped together to form large-scale functional units inside the plant.
And honestly, it's pretty straightforward. There are only 3 main tissue systems. First, you've got the epidermal system.
This is the plant's skin, its protective outer layer. It includes stuff like the epidermis itself, the stomata for breathing, and even root hairs. Second is the ground system. This is basically all the filler tissue. It's everything that isn't skin or plumbing. Think of the cortex and the pith. And third, the vascular system. That's the plumbing we just talked about, the transportation network made up of xylem and phloem.
All right, now for the main event. We've got our building blocks, we've got our blueprint, so let's see how it all comes together to actually build the different parts of the plant, the root, the stem, and the leaf. This part is so important for your exams because we are going to be comparing dicots and monocots side by side. Let's start from the ground up, with the root.
Okay, let's draw this out. Imagine we're slicing through a dicot root. On the very outside, you've got the epidema, with these tiny root hairs sticking out to absorb water. Go one layer in and you hit the cortex, which is a big zone of parenchyma cells. The very last layer of the cortex is the endodermis. And this layer is a huge deal because it has these things called Casparian strips. It's like a waxy, waterproof gasket that forces water to go through the cells, not around them. It's a security checkpoint. Inside that, you have the pericycle, which is where new lateral roots sprout from. And then right in the middle is the stele, with the vascular tissue. You'll typically see 2 to 4 bundles of xylem. And the pith, that very central part, it's tiny. Sometimes it's not even there.
Now for the monocot root. At first glance, you might think, hey, this looks pretty much the same. And you're right. It's got the epiblema, cortex, endodermis, pericycle. The basic layout is identical. But here are the key differences you absolutely have to know. Look at the vascular bundles. Instead of just a few, they're usually more than 6 xylem bundles. We call this a polyarch condition. And the second huge difference, look right in the center. See that big, well-developed pith? That's classic monocot root, totally unlike the tiny pith in a dicot root.
And this slide right here just sums it all up. This is pure high-yield exam fuel. Dicot root, 2 to 4 xylem bundles, a tiny pith, and it's capable of secondary growth. Monocot root, more than 6 or polyarch xylem bundles, a big obvious pith, and no secondary growth. Seriously, memorize this table. It's a guaranteed source of questions.
Okay, let's move up the plant to the dicot stem. Again, we start on the outside with the epidermis. Right underneath that is a layer of collenchyma for flexible support, then the cortex. But the real star of the show is the vascular bundles. Look at how they're arranged in a perfect, neat ring. Each bundle has xylem and phloem together, and they're called open because there's a little strip of cambium sandwiched between them. That cambium is the secret to secondary growth. And right in the very middle, you have a big, clear pith.
The monocot stem, though, is a whole different story. The first thing you notice is chaos. The vascular bundles aren't in a nice ring at all. They're just scattered everywhere throughout the ground tissue. There's no clear cortex or pith. The layer under the epidermis is made of tough sclerenchyma. And the vascular bundles themselves are closed. There's no cambium, which means no secondary growth. You can also see that the bundles near the edge are smaller and the ones in the middle are bigger. This scattered look is the #1 telltale sign of a monocot stem.
And here we go, another critical summary table, this time for stems. Let's hammer these points home. Dicot stem, bundles in a ring and they're open. Monocot stem, bundles are scattered and they're closed. In dicots, the ground tissue is nicely differentiated into a cortex and a pith. In monocots, nope, it's all just ground tissue. Monocots also have a tough bundle sheath around their vascular bundles. And remember our old friend Phloem parenchyma? Present in dicots, absent in monocots. This is another one you've just got to know cold.
Last but not least, the leaves. A dicot leaf has a clear top and bottom. To cut down on water loss from the sun, most of its stomata are on the shadier lower side. And if you look inside, the mesophyll tissue is beautifully organized into a top layer of tightly packed palisade cells for photosynthesis and a bottom layer of spongy, airy cells for gas exchange. Now contrast that with a monocot leaf. It's basically the same on both sides, with stomata on the top and bottom. And the mesophyllus isn't differentiated. But here's a really cool feature in grasses, these big empty bulliform cells. When the plant is short on water, these cells go limp, which causes the leaf to curl up. And why does that matter? It's a genius way to reduce the surface area exposed to the sun and save water.
All right, we've talked a lot about primary growth, how plants get taller. But you know that many plants, like trees, also get wider over time? This increase in girth is called secondary growth. This is a super important topic, and I know it can seem a little complicated, but trust me, we're going to break it down into simple, easy-to-follow steps. It all comes down to those lateral meristems we mentioned way back at the beginning.
So,
The very first thing that has to happen for secondary growth is the formation of a complete ring of vascular cambium. How does that work? Okay, remember those open vascular bundles in the dicot stem? The little strip of cambium already inside each bundle is called the intravascular cambium. That's part one. Then, some cells that are between the bundles become meristematic and form the intervascular cambium. This new tissue connects all the little strips together, forming one continuous ring. So the simple formula is intra plus intercambium equals the complete cambial ring.
Once that ring is formed, it gets busy. It starts dividing, making new cells. It produces secondary xylem towards the inside of the stem and secondary phloem towards the outside. And it makes way more xylem than phloem. In places with seasons, this creates annual rings. In the spring, when there's lots of water and sun, the cambium makes wide vessels to move a ton of water. This creates a light-colored springwood. Then in autumn, things slow down, and it makes narrower, denser vessels, which looks like a dark ring of autumn wood. One light ring plus one dark ring equals 1 year of growth. That's how you count the age of a tree. Now, over the years, the old xylem in the center gets clogged up and stops working. This becomes the dark, dense heartwood, which is mainly for support. The outer, lighter-colored, working xylem is the sapwood. It's the part that's still transporting water up the tree.
Phew. Okay, that was a ton of information, but you've made it through all the core concepts. So now, let's switch gears a bit and really focus on getting you ready for your exams. I'm going to throw a typical question at you, and then we'll do a super fast recap of the most important points from the entire chapter. All right, here's a classic question pulled right from the concepts we just covered. What is the function of the Casparian strip in the endodermis of a root? Take a second. Think back to that root diagram. What was that waxy layer doing? And the answer is, it's a checkpoint. Because that suberin layer is waterproof, it blocks the easy path between cells and forces water to go through the cell's membrane. This gives the plant total control over what minerals get into the vascular system. It's a brilliant little biological filter. Okay, time for the final rapid-fire recap. Let's lock these in. Remember, apical meristems for length, lateral for girth. Parenchyma is living and for storage, sclerenchyma is dead and for support. Endarc xylem in stems, exarch in roots. Open bundles in dicot stems for growth, closed in monocot stems. And the big picture, the neat ring of bundles in dicot stems versus the scattered mess in monocot stems. If you can master this internal architecture, this incredible anatomy, you've absolutely got the key to understanding how a plant truly works.